So as we get into the nuts and bolts of the discussions of anatomy, let's start with the cells and the tissues of the body. This is going to be where we start our systematic uh, study and integration of the functionality of the body itself. What we have to do first, we have to go back to our general biology and remember our generalized cell structures. And we're going to be looking at animal cells because that is the cells that are going to be making up the cells and the tissues of our body. So we'll start with this wonderful schematic of the generalized cell structure. Now, it's generalized cell structure, which means that some cells will have some of these uh, subcellular organelles and some cells may not. All cells, at least during the development of the cell, will have a nucleus. The nucleus is going to house the genetic information. In this nucleus, it's surrounded by what's referred to as the nuclear envelope, which is a phospholipid bilayer. Central to the nucleus is the nucleolus. The nucleolus is going to be housing RNA as well as ribosomes and ribosomal RNA that the cell does not necessarily need at that moment in time. Within the nuclear envelope, within the membrane surrounding the nucleus, there are small openings, the nuclear pore. The nuclear pore is large enough to allow for ribosomes and RNA to move from the nucleus into the rest of the cell, the cytoplasm. However, it's not large enough to allow the DNA to move out, which is going to get to what we're going to talk about here in a few minutes here, the genetic machinery of the cell itself. Surrounding the cell, we have a membrane unit. The membrane unit is the endoplasmic reticulum. This endoplasmic reticulum will have different names based off of the type of tissue and type of cells that we're talking about. Sometimes this endoplasmic reticulum will be referred to as the sarcoplasmic reticulum in muscle and a neurolemmic reticulum inside of neurons. The endoplasmic reticulum has two types of endoplasmic reticulum. One that has ribosomes attached to it, that is the rough endoplasmic reticulum, and then one that has no ribosomes attached to it, and that would be referred to as the smooth endoplasmic reticulum. The endoplasmic reticulum is responsible for production of materials for the cells, whether they are lipids, smooth endoplasmic reticulum, and rough endoplasmic reticulum, or proteins, rough endoplasmic reticulum. One of the organelles that most of the cells within the body have are going to be mitochondria. Mitochondria are responsible for the aerobic metabolism of the cell. Tissues that are aerobic tissues will have a higher amount of mitochondria than tissues that are anaerobic tissues. Tissues that are aerobic obligated tissues will have a larger amount of mitochondria than tissues that just are normal glycolytic tissues. In order to move things in and out of the cell, we have to somehow embed them into and take away membrane units. The organelles responsible for the production of these membrane units is what's referred to as the Golgi apparatus. The Golgi apparatus is sometimes also referred to as the Golgi discs and is sometimes referred to as the Golgi bodies. The Golgi apparatus, Golgi bodies, or Golgi discs are all going to be building membrane units from the materials that are being built inside the cell by the ribosomes or the endoplasmic reticulum. These will then vesicillate, turn into vesicles, which will then be able to embed into the membrane to go ahead and exude exocytotically release into the interstitial spaces surrounding the cell. Other prominent organelles that we will see include things like lysosomes and peroxisomes, which are responsible for the digestion of things within the cell itself. The peroxisome will also be responsible for storage of fats and lipids within the cell itself. There are other types of vesicles referred to as the microbodies. One of the other types of microbodies that are predominant in cells is what's referred to as the vacuole. The vacuole is principally a storage site for materials. We will see these vacuoles in high detail within the adipose tissue, where the vacuole will be there to store triglycerides, lipids, within the adipose tissue itself. We then have a whole bunch of protein structures. Protein structures are going to be there to keep the cell in a structural integrated unit. These include things like cytoskeletal proteins. Cytoskeletal proteins are made up of two predominant protein structures, the microfilaments and the microtubules. We'll talk more about the microfilaments and the microtubules as we talk about how muscle functions and how neurons function. And then we have the centrioles. The centrioles are cytoskeletal proteins that are responsible for division of the nucleic acids during the cell cycle itself through the production of what's referred to as spindle fibers. Surrounding the cell itself is a phospholipid bilayer, which we'll get more detail about here in a little bit, and that is what's referred to as the cell membrane. There's an integration in between the cytoskeletal structures and the cell membrane, which lead to the formation of what's usually referred to as extracellular projections. These include things like the flagellum. The flagellum in humans is only seen in one distinct cell in one gender, and that would be the sperm cell for males. That's the only cell that we have that is distinctly human that has a flagellum to it. We then have smaller projections, which are referred to as cilia. Cilia is usually numerous. We will see them on what is referred to as ciliated cells. The cilia and the flagellum allow for the cell to move. Now, in the human body, most of the cells that have cilia are linked to other cells. And so what ends up happening is that instead of the cell moving, it's the medium that surrounds the cell that will move when the cilia are functional. The other extracellular structure, which doesn't allow for motion, but what it does is it increases the surface area of the cell, and that's the microvilli. Microvilli we'll see in cells that are responsible for absorption or excretion of materials, where we're increasing the surface area. We're going to talk about this increase in surface area here in a second when we talk about the cell membrane, but there is a distinct advantage to having more surface area if I want to be able to exchange materials with the surrounding environment. And so what the microvilli does is for cells that have these microvilli is it increases how much cell surface I have to get things into and remove things from the cell.